Kia ora, hello, my name's Jovarn Sullivan. I'm a PhD student at the University of Canterbury in New Zealand, and today I'm going to be sharing with you some of my PhD work on myrtle rust. So what is myrtle rust? Myrtle rust is a plant disease caused by the biotrophic rust fungus 𝑨𝒖𝒔𝒕𝒂𝒂𝒖𝒄𝒄𝒊𝒏𝒊𝒂. It was first found in Brazil in the 1880s, and since then it has spread through the Americas, across the Pacific into Asia and Oceania, as well as South Africa. It infects plants in the Myrtaceae family, which is one of the largest angiosperm families with over 5,000 species, and is at present seen to infect over 500 of these species, which is really unusual for a biotrophic pathogen, which are usually host-specific. It's characterized by these yellow-orange rust-like pustules that form on the surface of plants, mostly new leaves and shoots, and it's really devastating to these plants, and it's this repeat infection, which is usually the cause of death. So I'm very interested in the infection mechanism, and so I'm looking into effector proteins, which are used by plant pathogens to increase their fitness against hosts. So they do this to overcome basal plant immune system. So they want to suppress any immune response that the plant may be having to keep themselves incognito, as biotrophic pathogens want to maintain the plant's life so that it can steal its nutrients, so that they can complete their own life cycle. So several proteins have been discovered to be differentially expressed by 𝒑𝒔𝒕𝒂𝒂𝒂𝒂 during the first 24 to 48 hours of infection. And this was done by our collaborators, and they were inoculating Manuka, which is leptospermum scoparium, with 𝒑𝒔𝒕𝒂𝒂𝒂𝒂, and they were getting the transcripts from that. You might have heard of Manuka before from things like Manuka honey, which is a big export of New Zealand. The focus of this research is just on one of these effectors, which is AP1260, which is the one I will be talking to you about. So 𝒑𝒔𝒕𝒂𝒂𝒂𝒂 is a dikaryotic fungus, so it has two nuclei per cell, and this has led to the recent discovery of two haplotypes for this protein, one derived from each of these nuclei. I have done a lot of work on one of these two and not a lot on the other, but I will make note of where this is. Current methods are ineffective. At the moment, it's just fungicide spraying and monitoring the spread of disease. So we need to look for new methods. So this is why we look to effector-based treatments, because they have been seen to work with other plant pathogens. And why is this? Why is this important? Economically, it's important because of large exports in the forestry industry, like eucalyptus, and other things like Manuka honey and clove and allspice and guava, all very important exports for numerous countries. So a big economic issue. Ecologically, obviously, species being driven to extinction in the environment is never a good thing. And then culturally is a huge issue. In New Zealand, especially, the Māori people, the First Nations people of New Zealand, have a really strong connection to nature and the environment around them. And it's really important that we continue these connections between Indigenous people and their environment and the things that are important to them. So I've done a lot of bioinformatic and biophysical analysis. So bioinformatic analysis, I use the protein sequence to gain information and potential characteristics of the protein, such as sequence homology. We do sequence homology, we do structure prediction, and we do disorder prediction. And then we can use this to inform the biophysical analysis, where we need milligram quantities of protein to obtain its characteristics in solution. So we do this using Escherichia coli, E. coli. We overexpress the protein in this, and then we purify the protein from the E. coli cell contents, and then we can use that for our structural analysis. So for analysis, we use that pure protein, and then we do different techniques like analytical ultracentrifugation, circular dichroism, small angle X-ray scattering, and nuclear magnetic resonance. And then we can also do functional analysis, where we express the the effector construct in different systems and see what happens. So yeast to hybrid for looking for interaction partners, and then using agrobacterium to express in planta in the Nicotiana benthamiana in specific, so we can see where it localizes within plant cells. So some bioinformatics have been done on these two haplotypes. So as you can see, sequence-wise, between the two of them, there's not a lot of difference. There's 10 residues that vary between the two haplotypes. Some are quite irregular changes, others are like-for-like amino acid changes. However, in the predicted structures on the bottom left here, you can see it doesn't drastically impact the structure. They both have this globular region at the top and a tail at the bottom. The disorder prediction you can also see is predicting these proteins to be disordered from around residue 90 onwards, and that tail you can see does start at around residue 85. So biophysical characteristics, we use analytical ultracentrifugation, which is where we spin the proteins at 250,000 times gravity and we get sedimentation data from that. So we measure the absorbance as they sediment. And from that, we can get information about the mass and the shape of these proteins. And as you can see, they're quite similar. They're both 15 kilodaltons. They're expected to be 15 kilodaltons, which means they're monomeric, they're pure, and also we got a bit of information about the shape, which showed that they were asymmetrical. Circular dichroism is a light bending technique where we can get information about the secondary structure of proteins. As you can see here, both of them have relatively similar traces, slightly shifted, but they both have the trend of being a random coil or unfolded with a little secondary structure. So we're looking for the peaks here at where we get our minima, and we're getting minima around the range where we would see for random coils and a little bit of secondary structure like alpha helices and beta sheets. So small angle X-ray scattering is a technique where we use synchrotron light to see the protein's dynamics in solution. And so on the left here, we have a PFR plot, and the skewed to the right, it shows that is rod-shaped or elongated. The graph in the middle is a Kratky plot, and this is showing that we're partially flexible. It's not a perfect bell curve, and it doesn't skew off into the distance, so it's really showing that it is flexible and partially unfolded. And then on the right here, in the dark blue is our experimental data, and in the light blue is our predicted fit for our model. And so we can see that our predicted structure is actually quite similar to the actual experimental data, which means that our model is good. So nuclear magnetic resonance here, we use the magnetism difference of nuclei to see structural information. And so here you can see that the two haplotypes are relatively similar. There are some shifts in the peaks, which is expected from the amino acid differences. However, they are relatively similar, and you can see this big blob in the middle is indicative of a disorder, and then the very clear peaks around the outside are indicative of folded regions. So again, we're getting that partial folded character. Then onto the functional analysis, we've done agrobacterium-mediated expression in Nicotiana, not myrtaceous, so it will be interesting to see whether when we get permits to work in myrtaceous plants, whether this makes a difference. But as you can see, with this is haplotype 2, haven't had an opportunity to do this in haplotype 1 yet, but it is definitely looking to localize around the endoplasmic reticulum and the nucleus, which definitely could infer some function for it. Whether this will be the same when we do it in myrtaceous plants is yet to be seen. However, this is definitely a good preliminary result and definitely very interesting. Then yeast-2 hybrid, this is in the very preliminary stages. I don't have any results yet. However, how it works is you have a bait and a prey, and so we have our protein of interest and then a library, and if we get an interaction between two proteins, either our protein of interest and some protein in the library, we get growth of yeast. And so we grow on really selective media, and so you only get growth of yeast if you're getting a really strong interaction. And then you can take those and sequence them, and you can get your interaction partners. And at the moment, I haven't quite gone to the stage of sequencing yet, but it is looking like it's going to go hopefully quite well very soon. So AP1260, we found that it's monomeric in solution, it's asymmetrical, it's partially folded. The sequence variation between the haplotypes doesn't hugely impact the structure. The potential interaction in the endoplasmic reticulum and nucleus is really interesting, and that its interaction partners are still presently undiscovered, but well on the way to being found. So in conclusion, characterizing AP1260 drastically improves our understanding of ascidiae infection. We know that it is really devastating and it's really good at infecting, but we just don't know how. We can now inform strategies for biocontrol and disease management, and it's a really good step towards providing an environmentally friendly method of control that's not fungicide spraying. And this is really critical for the survival of already declining species. Just like to say thank you to my funding, and also thank you very much for the opportunity to speak.